Under normal physiological conditions, the human gastrointestinal tract operates on a strict pressure gradient. It utilizes highly coordinated peristaltic waves and specialized sphincters to move matter, propelling it strictly downward through a one-way system. Reversing this flow requires immense physical force. The body must halt the baseline peristaltic rhythm, overcome the resting tone of multiple sphincters, and drive matter upward against the pull of gravity. This upward trajectory forces acidic material past the trachea. Without instantaneous airway occlusion, pulmonary aspiration can occur, a lethal event that destroys lung tissue. These mechanical requirements and the constant threat to the airway require the central nervous system to coordinate a full-body emergency protocol. To understand the mechanics of this override, we must examine the anatomical hub of autonomic control, the medulla oblongata in the brainstem. This schematic isolates the medulla. Within its lateral reticular formation, the vomiting center exists as a neural network executing the emetic sequence. Here, the nucleus tractus solitarius acts as the primary data receiver, processing incoming sensory signals from the body. The vomiting center operates like a biological CPU. It remains dormant while the nucleus tractus solitarius evaluates threat signals, initiating the reflex only when combined stimuli breach a specific action potential threshold. The most critical stream of chemical threat data originates in the chemoreceptor trigger zone, or CTZ. On this neural map, the CTZ is located at the area postrema, positioned on the floor of the fourth ventricle. Its exact placement relative to the blood-brain barrier is a defining anatomical feature. The CTZ sits outside the blood-brain barrier. This allows it to sample systemic blood and cerebrospinal fluid directly, monitoring for circulating toxins, opioids, or chemotherapy agents. The CTZ maintains a dense neuroreceptor profile, including high concentrations of dopamine D2, serotonin 5-HT3, opioid, and substance P receptors. Placing this sensor outside the brain's protective shielding provides an evolutionary advantage. The body detects blood-borne poisons and initiates their physical expulsion before they can cross into protected neural tissues. The medulla also receives motion data from the vestibular system via the 8th cranial nerve, utilizing histamine H1 and muscarinic M1 receptors to detect equilibrium disturbances. This schematic tracks the additional pathways converging on the medulla. The vagus and glossopharyngeal nerves carry mechanical data from the GI tract, firing in response to physical distension or chemical irritation. When gastrointestinal mucosa is irritated, specialized enterochromaffin cells release serotonin. This binds to peripheral 5-HT3 receptors, triggering the vagal afferent pathway. Direct inputs from higher cortical and limbic centers also feed into the vomiting center, allowing severe pain or increased intracranial pressure to trigger the medulla directly. The vomiting center constantly aggregates these vestibular, peripheral, cortical, and chemical data streams. If a stimulus arrives that is severe enough to reach the threshold, the emetic sequence begins. Once the threshold is breached, the vomiting center fires motor efferent commands down specific cranial and spinal nerves to initiate phase 1, pre-ejection. Cranial nerves 5, 7, and 9 drive immediate hypersalivation. This saliva is rich in bicarbonate to coat and protect dental enamel against incoming gastric acid. This split screen tracks the motor response. Vagal efferents halt downward gastric motility and initiate a giant retrograde contraction, which sweeps upward through the small intestine to force contents back into the stomach. Simultaneous sympathetic activation causes the diaphoresis and tachycardia clinically associated with acute nausea. Phase 1 is a staging sequence. It alters the biochemical environment and shifts autonomic tone to prepare the body before any mechanical expulsion occurs. Phase 2, the retch, is the mechanical preparation phase. The body tests the muscular force required for expulsion. Airway protection is the absolute priority. Cranial nerves 10 and 12 elevate the larynx and pull the vocal cords together, tightly sealing the glottis. With the airway secured, the intercostal muscles and diaphragm begin rhythmic, spasmodic contractions against the closed glottis. During these spasms, the pyloric sphincter relaxes. This allows alkaline duodenal contents to wash back into the stomach, raising the pH of the gastric fluid. The retching phase builds kinetic energy inside the abdomen, while the closed glottis ensures the contents cannot enter the lungs. The body then transitions into phase 3, ejection. The spasmodic rhythm is replaced by a single, sustained, maximal contraction of the abdominal wall and diaphragm. The soft palate elevates to occlude the nasopharynx, while the lower esophageal sphincter relaxes entirely, clearing a direct path from the stomach to the mouth. A massive spike in intra-abdominal pressure forces the gastric contents upward through the esophagus and out of the body. This ejection is the biomechanical endpoint of the reflex. The central nervous system coordinates multiple systems to achieve the rapid clearance of a perceived toxin. This physiological response creates a significant clinical challenge in anesthesia. Surgical pain, volatile gases, and opioids all flood the brainstem with threat signals. This composite diagram shows the redundancy of the afferent network. A single class of antiemetic drug is insufficient. Blocking the 5-HT3 node leaves other pathways open. Stimuli traveling through unblocked H1 or dopamine receptors can still achieve the threshold to trigger the medulla. Effective clinical intervention requires a multimodal strategy. Combining different drug classes allows for the simultaneous blockade of multiple afferent nodes. Clinicians must outflank the redundancy of the brainstem's detection network. If even one pathway is left open, the emetic sequence can still execute. Emesis operates as a unified feedback loop, connecting the specialized nuclei of the brainstem directly to the abdominal viscera. The reflex coordinates highly sensitive neural sensors with a strictly timed sequence of somatic and autonomic muscle actions. Mastering the precise neuropharmacology and the pressure dynamics of this reflex allows medical professionals to effectively predict, intercept, and dismantle the engine of emesis.